everybody, and welcome to the first episode of Science. I am David Mankoff.
I am Brett Kaminsky.
Andrew Dubinsky.
And what is this on our screen here? Introduce yourself, sir. Somebody's speakers are going.
I'm retarded.
Hello, everybody. Yeah, it's okay. First time.
I'm retarded. I forgot I had the stream up.
That's awesome. Hi, everybody. Welcome to the first episode of Science. Again, we introduced ourselves. So who are you on the screen there, sir?
My name's Kyle Schachtschneider.
How you doing, Kyle?
Pretty good. How about you guys?
What's that?
Pretty good. How you guys doing?
We are good. A little
good.
fun technical difficulty. We actually started on time, I think, for one of our first shows.
We're
ever.
within a minute. We're
2 minutes.
Good.
2 minutes.
2 minutes? Okay.
I gotta
can live with
say,
that.
I attribute science to that.
Yes.
So this is a new show with Geekify TV that we're going to be running periodically. We're going to have new things, articles, and things going on in the science world. We all have a little bit of fun background in science. Me, I have my pharmacy degree, so that's pretty much science. Brett dabbles in science.
I have the science of advertising.
You have the science of advertising, and you
Psychology.
dabble. Like it's
I
more.
dabble.
It's
I'm more space-oriented, exploration, astrophysics-oriented
right.
than anything else.
And
though.
it's one of those big passionate things on your side, which you do. And Andrew definitely.
I'm just a mad scientist.
You are a mad scientist, making
evil genius
concoctions.
sort of thing.
He makes crazy drinks and all that fun stuff. And on our screen here, we have Kyle. I do not know how to pronounce your last name.
I will
I'm
butcher it,
sorry.
and I've known him for years.
Wait, I think I got this. Schachtschneider?
I'm not answering any questions unless you can pronounce it correctly.
Oh, no.
Oh.
It's a
Fail.
short show, then.
Oh, shoot. I can't hear you guys anymore.
You cannot hear us anymore? That's lovely. We can still hear you fine.
Really? Okay.
Yeah.
Oh,
Well, I can
how
hear-
did you answer us?
Yeah. Wait a second.
You now. Oh.
All right, so
we
how do
go.
you pronounce your last name, sir?
It's Schachtschneider.
Schachtschneider. Okay. That's an awesome German name.
Yeah.
I love that. I told you it was German.
Yeah, very much so.
Yeah.
All right, and tell us a little bit about yourself.
Well, so I'm a grad student at the University of Illinois. We study genetics in particular. We work with pigs. We try to do a lot of biomedical research, human disease models. We want all the stuff we do to have like an actual impact. Science for science sake is fun, but science that actually leads to something is a little more exciting, I think. So that's the kind of stuff we try to focus on.
Definitely. And what are you guys up to right now?
I'm sorry, can you repeat that?
Sure. What do you guys
Submission.
you guys are doing any current research like right this second? What are you up to right now?
Yeah, so the research that I'm currently working on, I just finished up the paper for my PhD. So I'm done with my research, which is nice. So I'm helping out now with a couple of projects at the lab. We're doing creating what's called an Epi Genome, which is it's a type of modification, DNA modification that can affect whether or not genes are expressed or not. It's a very common issue in cancers. They can have methylation patterns that are abnormal, which cause certain genes to be turned on, which cause the cells to become cancerous. So that's a project we're currently trying to get going. We also have a project we're working with a transgenic pig that we're hoping is going to be a cancer model that we can use to basically study pretty much any kind of cancer you can think of.
Now, my question for you is, I do love bacon, but why pigs?
I was going to say it, and you beat me to
I
it.
did.
I was gonna say a common question that we get asked a lot is whether we feel bad eating pork after working with pigs, and I can tell you the answer is definitely not.
Good.
But the reason why we focus on pigs is because they're readily available. You can find them in pretty much any state, and they're very similar to humans, a lot more so than mice, which is the more common animal model. However, mice there's a lot of breakthroughs that have happened in mice that just don't translate well to humans. So
Okay,
the
this is
hope
actually
is that
something I remember from the thing in Nature was that pigs difference in genes from mice to humans has a bigger variance than it does from humans to pigs. Is that correct?
Yeah, that's another. So I don't know the exact percent, but
it was like 2% to 4% or something like that. It was significant.
That's about right. Yeah, and the pigs are also obviously they're a lot closer to us in size. Their internal organs are around the same size as ours. So there's a lot that that makes them more beneficial as a animal model. The only reason why they're not the number one choice at the moment is just simply because of cost and ease. You can get a new generation of mice, within a couple months, whereas pig, you're talking a whole year. So that can really prolong your experiments, especially if you're trying to get done anytime soon.
Yeah, I think, in my opinion, the absolute best is just human himself.
Yeah,
and I know that obviously does not work.
Just human trials.
Yeah,
Let's just make clones and do it.
One day, science will catch up and do
we'll
that.
We'll have The Island. Isn't that
that
movie
great
with
movie.
What's her freaking name? I was just talking about her.
I know who you're talking about.
Scarlett Johansson.
Yeah.
And Ewan McGregor.
Scarlett Johansson in that?
Yeah.
I forgot about that.
Yeah.
So you see, we're not just science. We're gonna have a little bit more because we,
tangents will happen.
Exactly. Yeah, tangents will happen. Forgot to warn you about that part.
I did. I warned him.
Oh, you did. Okay,
Yeah.
good. And we can swear. Like, there's no don't feel bad.
I'm aware.
People swear in science.
I gave him the rundown.
Okay, good. So yeah, obviously humans would be the best. That would be amazing. But we can't do that due to many, many reasons. And I don't know if people realize
people have too many ethics.
They do. They
and
do.
morals.
It is really hard
none.
to do a study. I was involved in a few studies in pharmacy school, and dealing with the boards and trying to get approval for this and that is such a pain in the ass. There is so much work you have to do in order to just begin the research. And sometimes, so many times, you're like, can we just start this? Like, we could have solved cancer by now. Can we just do this study? And it's ridiculous. I'm sure you know, Kyle, all the bullshit you have to go through to get approval, to set everything up, to have all the variables set in place before you even start the experiment.
Oh yeah, I mean the red tape that we have to deal with is unbelievable. Especially at the university, we're a public university, so we're technically part of the state. So there's all sorts of different groups, both like at the state level and at the university level, that we have to get approval from. And I mean, you would not believe if you have to, if you have to do something as simple as collecting a fecal sample from a pig, you have to have a protocol as to how you're going to do it, and any possible thing that could go wrong.
Yes.
Why this is humane or not humane. I mean, there's tons of.
Don't you just grab a shovel
along with it?
No, you do not. Do you guys use? Is it NIH? Is that the criteria for it? I can't remember now. It's been a little while for me. But do you guys? Do you have a review board that looks at all of these?
Yeah, we've got one at the university. It's called IACUC, which is I don't know what it stands for.
Big
but
acronym,
right? It's similar to the like what you would have at the NIH. It's just an internal board, and of course most of them are not scientists. They're business people, so it can be quite irritating at times.
Well, you have to write it in a certain language. That what struck me the most is you have to word things a certain way, and we had at least.
I'm sorry. I'm having
some stream difficulties.
I just won't touch my computer.
Yeah, just don't touch your computer, Brett, for the rest of your life. Sorry about that. The internet, we are wireless right now. We probably should be wired.
Yeah.
Which
I told you last week.
Yeah, I know, but
if only we went somewhere today that.
I'm wireless too, so I'm sure I'm not helping.
Yeah, no, I mean, I think it's a little bit on our end because we're also streaming and recording at the same time.
You changed it to 720p, right?
Yeah, it's the same setting as it was before.
Okay,
it's actually streaming fine, it says.
Okay, good.
As long as I don't touch it. But what was I saying? Oh yeah, so I mean, you have to review everything just dozens and dozens of times. You have to change the wording, and this word has to be that, and it's really freaking frustrating just to get research off the ground. Plus funding, you have to get funding. That's key. Like you say, the business people are the ones reading it. They're almost like your investors. Mm-hmm. So it's really, really difficult to get funding off the ground just to start research in the first place. And And I know we have the article here that was published. Now, can you tell us a little bit about this article we have here? I'm going to put it up on the stream as well here for everybody to see.
So the article was actually, it started, and I don't even know when it started, but this project started even before I was in grad school. It's been ongoing for probably around 10 years now.
Holy cow.
Yeah, and it's interesting. So the article, the point of the article is that we were able to sequence the swine genome as much as they have for the human genome. Came out a little while ago. So the basis behind the paper is the genome, which was sequenced. Well, the pig. Start with the pig. So the pig is a Duroc pig, which is a specific type of breed. They're actually really good for, I want to say bacon. So
all pigs are good for bacon.
So that's a good one to keep in mind.
That is. I remember that one.
Do you get to eat the remnants?
Yeah. So the pig that we sequenced was actually at one of the farms at the University of Illinois, which is pretty exciting because people from all over the world want tissue samples from this pig, and they want to be able to work with it. So it's pretty cool. It kind of puts us at the center of the universe, more or less, when it comes to dealing with pig genetics, which I can tell you is not a huge group worldwide.
I was going to say, when did it become? Sorry to interrupt you, but when did it become common to who thought of the first use of pig in research like this? Because it's always been mice and other animals like that.
Yeah, I couldn't tell you when it first started, but it's been something they've been pushing for quite a while. I mean, that was part of when we did the genome sequence. There's two phases. You're using it for agricultural purposes. There's a lot of money that came from USDA and that kind of stuff to fund it in order to find genes that match well with body fat compositions and different meat qualities. And then on the other side, we've got the biomedical aspect, and that's something that's we've been trying to push for a while, but as I said before, it's difficult, and the main reason is just the amount of space you need and the amount of money it costs. I mean, you can imagine, a dozen pigs versus a dozen mice. I mean, you could do an experiment with a dozen mice in your bedroom, and good luck doing that with a bunch of pigs. So
yeah, it definitely there's more concerns with pigs. It's definitely going to be more expensive and more tedious.
Yes.
But I mean, from the basic science that I understand it, much more accurate, much more of a better picture. So it's almost like it might be worth it, instead of using mice and having some strange things happen with that, to spending a little bit more money and investing a little bit more and having the pig population used instead. So basically, let me get this straight then, kind of, because I did read through some of this article. Basically, you've mapped out the genome of this specific pig, correct?
Yes.
Okay. And
we've go ahead.
No, go ahead.
I was saying, so we've sequenced the whole genome, and then we've mapped out the placement of all the genes, or at least all the genes that that we know exist so far. I mean, you're always finding new stuff, but there's it's a full reference. You can actually get it online as well if you ever have any interest in taking a look at basically just thousands of pages of G's, C's, A's, and T's.
Yeah, I was going to say, like the Gattaca.
This book is boring.
Yeah, right.
That's pretty much all it looks like, yeah.
Now, did you guys compare the human genome? Like, did you splice them together and are like, what's similar, what's different, and cut out the difference and see what really is in the nitty gritty in the genes?
There have been some other groups on our campus who have looked into that kind of stuff. I want to say about, maybe 3 or 4 years ago, when we were first getting all this together before everything was annotated, they were able to take, somebody at the University of Illinois took the human genome and they broke it apart and rearranged it, and they were able to assemble the pig genome from the human genome, just changing the chromosomes and the placement of all the genes. So
Holy crap, that's crazy.
Yeah, it's surprisingly similar. I mean, I wanna say 98%, but I'm not sure. But it's surprisingly similar, considering you know how evolutionarily different we actually are.
Right, right. I mean, yeah, pigs are fascinating creatures. My favorite thing with pigs is
Bacon.
well, besides bacon,
Pork chops.
the number two is if you take, you know, the domesticated pig is different than a wild pig, and if you take a wild pig, or I'm sorry, a domesticated pig, and you leave it in the wild, after like 3 weeks, it will start to resemble a wild pig. You know, it'll grow tusks and hair, and it'll get much more aggressive. It's really fascinating how that works. We don't really know exactly how that works even. Like, how do they know? Like, how do they figure that out?
Yeah, exactly. How do they know that it's time to start turning that stuff on? I know it's really fascinating stuff. It's something a lot of the evolution is something that a lot of these people have been very interested in. So that's a similar aspect of it. Yeah,
They didn't evolve. God put them there.
Exactly.
I had
right?
to say it. I had to say it.
It was going to come sometime. On a show with science, you have to talk about that.
Yeah.
Wasted all my time.
Yeah, you're right. The answers are right there.
It's in the Bible, man. Noah saved them.
It's just magic.
Yeah.
So now is the thought process that you have this mapped out and you're kind of like proving that pigs are more closer to humans than mice are. Do you think that it'll catch on and that people will start to use pigs a little bit more, even though it's more cost prohibitive at times?
It definitely is catching on, but the major issue is cost, and also you look at groups like NIH, and it's a lot easier to get a mouse study or to get them to fund a mouse study a lot of times because there's so much that's been done in mice. They're so well studied that it's pretty easy to convince people with money that I'm going to be able to do this experiment. It's going to work and it's going to be important and all that. But it is something that we're starting to see more. There's a lot of labs that are starting to try to produce different lines of pigs, transgenic pigs, just like we do have in mice. Mice, we have certain strains that are more prone to cancer or Alzheimer's or different diseases like that. So we're trying. That's our current goal. Is we're starting to use transgenics to try to produce pigs with similar predispositions.
Right. Actually, our buddy who could not be here tonight, Kevin, works in a lab that uses mice, and it's a neuroscience lab, and so he was telling me about that they have pigs that they have this sort of brain disorder, they have this sort of problem with them, and it's interesting that they can do that. Extremely complicated, I assume. And they're going to be able to do it with pigs too, correct? Like using this genome complex, they can do the same thing. They can have like this pig has this problem and that problem, kind of the same way they do with mice. That's kind of the goal, right?
Yeah, exactly. I mean, because the genome, the great thing about a genome, and if you've ever done work with an animal that doesn't have a genome sequence, which is how I started, it wasn't quite ready yet. It's very frustrating because nobody knows what any of these genes look like. They have no idea. So if you're trying to study a certain gene, you have to find it, you have to sequence it, you have to determine where it is and what it looks like. But the beautiful thing about now having this pig genome is you can just literally just go online and type in the gene you're looking for, and there's the sequence, and you can start working from there, and it tells you the protein content and everything. So it's very helpful for that kind of stuff.
Hey, Dave, did you put the video back up of us, or is it still?
Yeah, no, I got
it. I told him.
Okay.
Yeah. What was I going to say?
Sorry.
It's gone. No, it's not. It's okay.
I just saw one of the comments. I was like, oh, that's a bad idea.
Yeah, just keep it up there the whole time. Mm-hmm. So, okay, obviously my background is drugs. So I know drugs, as they say. My hope with this is that they do so much research in mice. I would like to see it more go to porcine. I would like to see it. That'd be very cool to go the pig style.
Mm-hmm.
Yeah.
Because I think it would be much more accurate. Because there's a lot of issues, actually, with some drugs that they've tested in mice that are great. They move on, and then it does massive amounts of liver and kidney damage to humans, and we're like, whoops.
Oh, you have cancer. Oh, this drug gave you AIDS. I'm sorry.
Exactly. There's a lot of issues like that we could help to eliminate a little bit more going the pig route. And when I first saw this, I was like, well, holy crap, I've never heard of that. Like in school, it's always mice this, mice that, mice to humans. And when I heard that, I thought about it for a few days, and I was like, that's actually pretty smart when you look at the organs alone, too. I know that the size and placement is very, very similar.
Yeah.
With humans, much more than mice, for sure. Especially like the size-wise, you know, like it's so it makes more sense when you have something as close as you can to a human without actually using a human just yet to do a lot of this research. And I think it's a great project too. I think it's really like on more of the forefront for the research industry, because it's like they're using mice, and if there's a better thing, you kind of have to crack that shell open and say, no, we have this better way to do it.
Yeah.
Like it's a little bit more expensive, but we will
in the long run, it's probably less expensive.
Likely we will get better results, which will lead to more profit, which is in the end what a lot of the investors want to hear.
Yeah. Money? What?
Yeah.
Where?
Exactly. And that's the big thing nowadays. And that's part of the problem we had with a couple of the studies I was involved in, is they didn't get quite off the ground when I was involved in them because of funding and all the other bullshit that kind of goes along with that. I don't think people realize how, like the article we have here, how painstakingly retarded it is to get that article together.
Oh, I bet.
Like all the pieces that have to put together, all the funding, all the typing and editing.
I feel like every 3 words was a link to another reference within the document. And I was like, what?
And so that's why I, like whenever I hear that when Brett told me you were published, I was like, wow, that is an accomplishment. Like that is one of the things that you're like, holy crap. I mean, I am not published because the studies I had to back out of because I graduated. So I do it for friends who were published, and it's a big deal. Like in the science community, at least, it's a pretty damn big deal to be published like that. Yeah. Very impressive.
It's exciting. I was really happy, and this article, like I said, this one wasn't my project, but it was one that I was a part of. So it was very exciting because this is the first one that I've ever had with my name on it. The paper for my PhD is getting published, I think at the end of the week, actually, coming up pretty close. So that one I'm really excited about because that one I actually wrote.
Yeah, that's like your baby. Can you talk about that at all?
Yeah, sure.
Okay.
So what journal are you in now for this one?
It's so the journal we published in, it's called PLOS One, which PLOS stands for Public Library of Science. And it's an online journal, so it's open access.
Oh, nice.
So once it's published, anybody can go look at it. The Nature one, I think it's open, but it's only open because it's a special article. They're usually subscription only. So
that's a great pun of a name, by the way. +1.
Yeah. So
way to derail them.
Yeah, sorry. that's my job here. I just derailed the conversation.
Okay, sorry. So my project, we study, again, going back to the biomedical aspect of it, we're looking at the effects of the bacteria in your gut, how they affect your immune responses. So what we did is we had a group of pigs that we took from their mother, and we raised them in research units at the university to kind of control their environment. And we exposed some of them to like a normal, healthy gastrointestinal tract, and then the other ones we kind of left alone, and and see how the early exposure to the microbes affects the immune response. And what we saw is that the pigs have a much stronger immune response and a much lower severity of infection compared to the control pigs, if you were, that were just kind of left alone.
I'm sorry. So what did you do to the experimental group with the? How did you change their GI tract?
So I was trying to kind of glance over that part.
No, yeah. Sorry,
so
I'm just curious.
Yeah, we administer them what we call a non-pathogenic oral inoculum, which is literally just pig shit mixed with lots of water, basically.
Right. It's kind of like a fecal transplant.
Yeah.
Like,
exactly.
I remember when we read about that not that long ago.
It's the same basic idea. Yeah.
And then is this fecal transplant something that you guys, is this the fecal matter, was that something you guys made, or was it something not you made personally?
Yep.
Yep.
Into a glove and into the pit.
Yeah, right.
Somebody made it. No.
Yeah, right. So what was the like? How was that made?
I wouldn't want that job.
It's literally it's feces from what we deem to be a healthy boar, out at one of the farms here at the university. So the farm is free of any major pathogens. It's considered a high health herd, which is kind of like top,
Right.
healthy as you can be as far as grading goes. And we literally just collect fecal samples from them, and we mix it with what we call PBS, which is basically just sterile salt water, like the fluids you get in an IV, and feed it to them with a syringe.
Cool. I bet that tasted
And
awful.
Well,
they're quite
pigs eat
frustrating to
everything.
feed because they absolutely hate it. They will not eat it, as you can imagine, but
Who would voluntarily
pigs
eat
do
shit anyway?
eat feces normally. So as you're trying to get the one to eat it, and it's spilling out, all the other pigs are right underneath them, licking it up off the ground.
Now, why is that? Why do they eat their own shit?
Wouldn't you?
If you
What's
were
that?
starving, wouldn't you?
I understand, like dogs eat they eat grass and stuff like that, and sometimes they eat their own poop as well because there are extra nutrients in them.
Mm-hmm.
Is that kind of the idea with pigs as well?
Yeah, it's same type of idea. It's a very common practice for them, which is probably part of the reason why we got it approved.
Right.
But yeah, it's totally normal. It's kind of just part of their nature.
Right, because I know for us it's like, ooh, gross, eating your own poop. But I mean, a lot of animals in the animal kingdom do that.
Oh,
it's a great way to save nutrients that you would normally lose.
Dakota does it all the time still.
Right. Yeah. She's
All
still
the
young.
time.
I mean, especially a growing puppy will do
Yeah.
that. Because they
Slowly
somehow
stopping, but she still does it. It's
Right.
disgusting.
So let's go back because it's okay. For people to know, it's normal flora, right? Is basically what you guys are studying.
I'm sorry. Can you say
The normal flora, like, is that kind of what you guys are studying? Like the flora, the bacteria in your gut. That's kind of
Yeah.
Okay.
So we're kind of we're focusing on kind of something. It's similar to this hypothesis called the hygiene hypothesis.
Yes.
You know,
I'm familiar with that actually.
Yeah. So it's basically that same idea as, if you have more of the if you're exposed to more bacteria early on in life, your immune system is more prepared farther down the line.
It's one of the theories that, for you guys who know, that the reason we have so many people allergic to peanuts is because
We sequester them.
is because of a lot of the use of like antiseptic and stuff like
Mm-hmm.
that. So there's something that we're not being exposed to in the womb, or we don't even know where yet. There's a big theory that with peanut allergies and a lot of other allergies actually.
It's
that
because we
kids have.
shelter them from everything.
Yes, and
Most people who are like our parents' age aren't allergic to peanuts. It's a newer thing because people are all like, oh, don't let them do this. And that time it's like, oh, go outside and eat dirt.
right.
It never happened for me. It's like my parents were like, yeah, just eat it. Okay,
Mm-hmm.
ate it. Like donuts fell in the mud. Yeah, it's fine. Just brush it off.
Right,
Mhm.
and your body needs that. That's kind of why you have an immune system. It's
Yeah.
kind of like it needs to be used a little bit.
And that's why, me personally, I barely ever get sick. But when I get sick, I am really, really sick.
And you bounce back pretty quickly usually.
Yeah. Like, what was that? A few weeks ago, I got sick on a Tuesday, and I was fine by Thursday.
Yeah, and there's some people who are sick all the time. We have a few friends who are sick all the time, and it's really funny. If you have a lot of other really, really bad issues, they could, we've talked about the human fecal transplants that they can do between family members.
Oh,
and
yeah,
stuff
that was
like
on IO9.
Yeah.
Several. We talked about
Yeah.
it on 3 Guys 1 Cup.
We actually, we did. We talked about it in school as well, because it's gross, but it's like you do need some bacteria, especially in your gut. I think our gut bacteria. That's how we get a lot of nutrients. We get our a lot of vitamin K, a lot of other stuff that we absorb through from the bacteria in our gut. That's why when you take antibiotics and you can get sick and have diarrhea and stuff like that, that you have so many problems. Where was I going with this? Oh, so my question was, so I got a little off track. It's been a long day.
Poo.
Poo. So poop. No. So, and is this? My question was: Is this with was this with younger pigs, or what was the age group for these?
This was with young pigs. So we got them at birth. It was actually this whole process was kind of long. It's nice, it's fun to get out of the lab and play with the pigs, but after a while, you definitely get a little sick of it. It's a lot of work. We had somebody watching the gilt, the mother, for about 48 hours before she was supposed to give birth, because we wanted to remove them immediately. We didn't want them to have any exposure to the mother's feces or allow them to suckle or anything like that. So we pulled them away immediately at birth, and we would hand feed them colostrum and then a medicated milk replacer for about every 2 hours for the first couple 3 weeks. And then we actually did the fecal transplant, if you will, at about 5 weeks of age. So they're pretty young. You know, a mature pig, mature like market ready, or one that's ready to start potentially producing, is anywhere from like 6 to 8 months. So it kind of gives you a little bit of a time frame on there. So they're pretty early. It would be the equivalent to within the first year for humans, I think. And then the experiment continued on till they were about four months old. So very young. But yeah, by 4 months old, I think they're all about 100 pounds, and it's
Holy
pretty.
shit, that's a big piggy.
Yeah. And,
That's a lot of bacon.
The problem is they're crazy smart too. So you start, you know, if you've ever seen, if you got to restrain a pig, you use a snare. If you guys have ever seen that before.
Yes.
Yeah.
And they're smart. I mean, after the second time you do it, they know what you're trying to do. So eventually you're just wrestling with them to the ground.
Right, they're not all the dumb animals, you know.
So what you're saying is you can go into being a rodeo clown?
Yeah,
This doesn't
pretty
work
much,
out?
right?
Have you ever greased them up and just tried to catch them?
The greased up deaf
What's
guy?
that?
Yeah. Have you ever just tried to grease them up and try to catch them?
I haven't tried to grease them up, but I've spent plenty of time chasing those damn things around.
Yeah, right, but you're sick of that.
All right, now,
I wouldn't even imagine trying to grease them up and trying to catch those things.
now
That'll be
correct
the
me
next
if I'm wrong. Or I thought you told me before that these were cloned pigs, or they're just naturally born pigs.
These ones were naturally born, but we did originally try to do the experiment with clone pigs. Actually, the clones were clones of the pig that we used for the genome sequence.
Gotcha. Okay.
The same one. So we did try it first. The problem with the clones is the technique is still just very has a very low success rate, like less than 1%.
Gotcha.
So what ends up happening is you put about 100. I think we put about 120 embryos into one pig, and he ended up giving birth to 4, I think.
Wow.
And
That'd
the
be
second
insanely
time-
bad if like half of them took by some miracle of just design all of a sudden.
Yeah, right.
She's like, oh, that's not good. That pig's gonna blow up with babies.
Oh man, I'd be more than happy to have 60 of them, man.
Yeah,
I'll tell you.
right. That is crazy. That is fascinating. I mean, normal flora in humans is very important, and that's an ancillary thing that is not recognized by a lot of people. It's starting to be. Definitely pharmacists are. I mean, we have a lot of probiotics and stuff like that over the counter and things like that. It's fascinating to see that. So there was a significant change then between the. I don't know. Can you spoil the study? Is that does that happen
no.
with you guys?
It's already been accepted. It's going to
Okay.
be published in minutes, so it's. Or,
I don't know if
you
you're like I can't talk about it till it's released. You know, or anything
It's like, well,
that.
shit. Maybe we should have done this last week.
No, it's too late for you guys to steal it. Good luck. It's already been through.
Because I know we did a ton of study stuff, so I know all like p-values and all that fun shit. So don't feel free to just lay those down, because we can explain them to everybody. If anybody else, if you guys have any questions, definitely interfere, because it's complicated stuff doing study like that.
Yeah.
So it was significant from I'm sorry, you're measuring again, because I don't have it in front of me. I'm one of those that I read a study like many many and highlight portions of it. That's how we were taught. So I always am like, what are you doing now? So I apologize if I'm like, what did you do with this now? These are pigs? No. If you when you guys did like the final results of it, you were measuring against the control. What were you measuring with the control?
So we would look at a range of two categories basically. Well, we would either be looking at immune responses or severity of infection. So for severity of infection, what we did is we infected them with a basically it's called mycoplasma. It's like a respiratory infection. It's very common in pigs. I'd say probably about 90% of pig farms are chronically infected. It just kind of gives them a cough. It's nothing too severe. But so we infect them with that, and we measure like coughing scores, the amount of times that they cough in a half-hour period. We also look at antibody production, how quickly they start producing antibodies against the mycoplasma. We looked at the lung lesions after they were euthanized. cytokine levels, and let's see, I'm forgetting something.
Was it white blood cell at all? Did you guys look at that, like immune response?
We originally looked at white blood. We originally we pulled white blood cells because we wanted to look at methylation patterns, but we weren't able to find anything. Although something that's not in the article but is kind of interesting, when I was extracting the white blood cells, when the process that you do it, you end up with two different extracts, and one of them has like basophils, eosinophils, neutrophils, all the
right, the usual players,
all the usual white blood cells in one pellet. And as the study went on, after the infection, and you could see the pigs were starting to fight off the infection, you could see increases in certain levels of white blood cells, which weren't significant because you could see it in both groups. But it was still kind of a cool thing to see, something that didn't make it into the paper, but it was kind of interesting.
Right. I mean, it's very important that you separate, because there's not just one white blood cell. There's many different types, and each one means a different thing. Where it's fighting, what it's fighting. It's kind of a good marker that we use a lot.
Yeah, exactly.
It's very cool that you can. It's very cool that you can do that with pigs. That you can have the same kind of a kind of idea. You can use the same lingo, kind of like.
Yeah. I mean, the great thing about biology, and especially genetics, is it's so translational. I mean, I could hop into a new job in a year and start working on humans, or work with mice, or any animal you want, pretty much. And you just have to learn the system, that animal, what their intricacies are. But otherwise, the actual science, I mean, it's all the same. It's nice. You can kind of hop around wherever you want.
Right. You can still talk the same lingo with everybody, like you and I are doing.
Yeah.
So what?
Yeah,
what?
exactly.
So now that the research, yeah, right. So now that the research is done, what's the next step for you?
Well, for me, I've got two more things I got to do before I graduate. I have to write my thesis, which is basically the paper that's coming out, plus a lot of the work that went into it that didn't make it into the paper, and some literature reviews, that kind of stuff. And then I have to defend, which is kind of like it's like a presentation of my work, and then basically people can show up and ask me questions about it, and try to make.
You're in a big room with one light bulb above your head and an audience all around you, and they're like, yeah, they grill you, don't they?
Yeah, you can't see anybody, and it's.
Yeah, I'm so glad I don't have to do dissertation. I mean, I had to take my boards, which is
honestly, that's probably one of the only reasons I decided not to pursue a science degree.
I
have no desire to be grilled by people that I just no.
Yeah, you don't know and probably don't like, right?
Yeah, exactly.
Yeah, that's like in the music world. Really quick, not to change subjects. In the music world,
but you did it.
Yeah, when you go for your major and stuff, you have the fuck's it called? Juries. That's what it is. And you're just up on stage. You got to go through all the different musical instruments, and each instrument you got a jury to go through.
Do you really? So it's kind of the same thing. Like
yeah,
you
just
kind of have to
exact
be,
same thing.
Yeah, you have to. They're trying to just like test your ability.
Yeah,
that's very
make sure you're qualified.
Very scary.
Yeah. Oh yeah.
Not a fun thing at all.
Yeah, it's intense.
So
that's
you're right.
So then I just took at the beginning of the month, I took what we call our preliminary exams, which is basically that one where you just they pretty much just sit you down and they ask you whatever they want about anything, and it's pretty intense.
It is, yeah. You definitely
preparation for that was not fun.
No, it's not. It's similar to the boards I had to take and stuff like that, where you're just. Although thankfully we don't have a live portion anymore. All of ours is on computer, because live would be very scary for sure. Definitely a bundle of nerves then. So how you said you had that part already? You had the first part?
Yeah, so I had the first one at beginning of December, which I passed. And yes.
Congratulations.
So that's pretty much that's the big one, which is nice. The defense is more. I mean, you have to go in there and you have to know what you're doing, but it's a lot more of a formality than the prelims. The prelim is the big one where they kind of decide if you're ready or not. So
right.
It's a little bit of a weight lifted, which is nice.
Yes, definitely. You can relax a little bit and then prepare for that. Now, when is that final push?
So it'll be, well, there's no date set yet, but it'll probably be sometime in April. It's about a month before you graduate, and I'm planning on graduating around May or June. Just kind of depends on how the thesis goes. and how many times my advisor sends it back to me.
Yeah.
So
There's a lot of writing and rewriting, isn't there, with that?
what?
There's a lot of writing and rewriting with the thesis.
I'm sorry.
There's a lot of writing and rewriting with the thesis.
Yeah. Yes. Sorry, I can hear you better for some reason. Yeah, there is.
I think it uses the mic off my laptop. I think it's just the mic is super sensitive on this.
Oh, okay.
It didn't make sense to me how our mics connected to your computer were linking to mine for some reason.
You know how it works? It works by science. That's
magic.
Science.
I thought it was all religion.
Yeah, magic is just science we don't understand yet. So let's go with it.
Exactly. Well, that's good. So you're almost to the end now. What? So you graduate. You get your big PhD. Your PhD's in, is it genomics?
It'll be it's in animal science is the
Animal
degree.
science. Okay. What do you want to do with that afterwards?
So what I really want to do, I want to, at least short term, I want to get a postdoc somewhere. I'm really interested in trying to find something over in Europe. So that's what I've been working on lately. My wife Lisa is art history major, so she really wants to be in Paris. So
I bet.
I'm working.
Now, what would you? Why Europe? What would you want to do over there?
I would like to be somewhere new, and it's probably the fact that I did my undergrad as well as my graduate school at U of I, so I've been here for almost 9 years now.
You've been in the sticks for far too long.
I started in 2004. Yeah, so just about 9 years. So it's I want to get out of here so bad they got to leave the country. Is basically
I don't blame you. Yeah, I don't blame you. Pharmacy school for me was 8 years, and most parts of it were not fun.
Pharmacy school is 8 years, or just your undergrad?
To get to the end, 4 years of undergrad and 4 years of the degree itself.
Gotcha.
And at the end, yeah, I understand that feeling. At the end, I was like, I want to leave for a while and not see and not think, you know.
Yeah.
That's great.
Time to get out.
That's very exciting for you.
That'd be a hell of a time lag for us to do the shows with you overseas.
Yeah, right. Could you imagine the internet lag there?
Yeah.
No, I'm glad I stayed here.
Oh, I know. It's gonna be like I'm gonna be getting up at like 4 a.m. to do this stuff.
Definitely.
You were just in the Netherlands for a conference.
Getting off topic, I had the hardest time. I was over in the Netherlands.
There we go.
The week before our playoffs, for fantasy football, and I was trying my heart out to get my team ready, but I couldn't. The Thursday night game was at like 3:30 in the morning for me, and I couldn't. I was trying to decide whether or not to start McCoy. I can't wait till game time. Oh, it was frustrating.
Yeah, that is actually kind of shitty if you think about it.
Yeah, that would suck.
Now you say a pain in the ass. Like, not great, you know.
You were in the Netherlands. What were you doing over there?
I was in the Netherlands for a week for a conference on sequencing, how to analyze sequencing data. This what we call next generation sequencing data, which is all the new technologies that have allowed us to start sequencing like the pig genome and everything in such a short amount of time.
Excellent. Was it so? How many wild and crazy parties were going on over there?
None, unfortunately. It was tough because it was we were not in Amsterdam and we were not.
That sucks.
A night, but we were in small town called Wageningen, which is it's literally just a university, and they call it a university town. But just to give you an idea, U of I, Champaign-Urbana is a university town, and we've got I think 40,000 students. And their university had about 6,000. So it was
university
quite tiny. Everything closed at 6 o'clock. There was one bar that was only open past 10 o'clock on one of the nights. It was, but it was good. It was cold, but I learned a lot, so I guess that's good.
You're from Chicago. The cold shouldn't bother you.
Yeah, that's true.
Yeah.
It gets very chilly here, although not so much this winter.
Yeah, not so much this winter. No,
it's been kind of mild here.
Same thing with last year.
Yeah, last year was a very mild winter. We're very, very lucky in the Chicagoland area this year.
I want the snow.
No, you do not. Fuck
Yes,
you.
I do. I want Snowmageddon again.
No, you do not.
Yes.
Why, so you can get off work or something?
Yeah.
Exactly.
Yeah, but it's just so much cleanup.
That's fine.
Oh, man. I was going to say something, but it's gone out of my head. What do you guys got?
You're the science man.
I am the science man. You know, it's funny. Kevin's gonna be kicking himself for not being on because his whole thing is studies in mice, and I'm sure you'd have a million different questions about pigs. I'm fascinated by that too. I was like, when I read it, I was flabbergasted for a few days. It was one of those things that sticks in the back of your head, and you think about it and you go, I like that. Like when you start looking at a lot of the research and stuff like that, you're like, I think it should go to that, even though it's cheaper for mice. There's a lot of issues we have with that.
Yeah.
I know that in Japan, I just read a study that they did in felines and cats about cancer study that I thought was really fascinating because I did not think they used cats as much because they had like a little bit. They thought it was a breakthrough, but you know when the media takes that and they spin it so much because the scientists were saying it's something positive, but they're like, well, wait. We haven't even tested this in like mice yet, or pigs for that matter. You know, so it's like, hold on now. But then the media took it and they're like, oh, there's a cure for cancer now. And
it was that one that came out of Canada about we have a cure for HIV, and it's like, wait.
Yeah, they're like, that's
they got
not
money?
Yeah, that's not what the science has said. You know, like they have something that's promising.
Like, it's something promising, and we're moving to human trials probably in the next decade.
Right. And it's like, so you gotta. Science is very, very slow, extremely slow.
A lot of that's due to government interference, but
and I mean, and funding too. It's slow for a lot of reasons. You know, even just the actual study itself, like it just takes months or years to do some things, and it's very frustrating. You know. As you know, Kyle, you probably know that firsthand.
Yeah.
You know?
Oh yeah. Well, that's part of the reason why it takes. You know, that's part of the reason why people use mice. You know, because it's quick and easy.
Right.
Well, I won't say quick and easy, but quicker.
Yeah, quicker and easier than using any other.
They're so
animal
readily
model.
available.
They reproduce so fast. It's almost not fair.
Oh,
you know, it's almost.
You can just go online and order them. Whatever you want. You want mice that produce tumors? You know, they got those. They got anything.
They do, yeah. Like you want this specific brain tumor or this specific, lung tumor? Boom, you can get this. Yeah.
Oh yeah, I got ones. Yeah, predisposed to certain ones. See, that's the interesting thing is that we've got all these different lines that do all these that are for all these diseases. But what we're finding out, and it's partly because of genetics, is once we start getting down to it, like there's an Alzheimer's. I believe it's also, yes, an Alzheimer's disease model in mice that people have been using, and they've been getting great breakthroughs in the mice. But what we're finding out now is that it doesn't translate to humans because the disease progression is completely different. It's different genes that are shutting down, and cells are dying in different ways than they do in like human Alzheimer's. So it's completely irrelevant when it comes to actual human studies.
I hate
people don't.
I hate those Alzheimer's drugs. They do not work. Like I have patients. Delaying the progression is so loose a term. You know what I mean? Like it doesn't. I feel like they're just water pills. Like they do not do what you think they're supposed to do.
Placebo effect is a very powerful thing.
Exactly, and
Here, have a sugar cube.
It's extremely expensive too. So all the time, like I see that exact thing because I know what you're talking about. That's what we talked about too in school. Was when they had that, they were like, they thought it was going to just revolutionize Alzheimer's. They come out with, Aricept and all these other drugs, and it's like, these are kind of crappy. Like now the studies looked okay, but then in the real world, they're like, oh, it's not really helping. It's not a cure. It's none of that.
Yeah, exactly. It just doesn't end up doing anything.
Right.
But it's interesting. You talk about like, people want to save money. They use mice because it's cheap and efficient or whatever. But then it's like, you look at it like that. It's like, how much money are you wasting on drugs that aren't going to end up doing anything for people, and maybe you'd be better off with a few less trials and using pigs or some other model.
Right.
Know?
Yeah, not only like the waste of money, but you waste so much time.
Yes.
With doing it on the mice, and then it's like, hey, pigs are more expensive. But
now, do you think that now that the genome is mapped out, that they will start picking up more porcine studies like this? Like they'll start doing a little bit more of this?
I hope so. I mean, I know in the private sector, at least, not the private sector, but in the universities, the research labs, it will absolutely help, and it'll make people more interested. And the question is the biotech companies, the pharmaceutical companies, whether they're going to start believing that this is something worth investing or not. And that's something I think it's still. We're gonna have to kind of wait and see.
And I feel like they almost want to wait and see a little bit too. Like as soon as one of the big companies really incorporates a lot of this pig research, then the other ones will, and if they have some success, like that's what it will take, is someone to open that door.
Yeah.
And then
nobody wants to be the first one.
Exactly.
And there's also, we've got, speaking of the Alzheimer's, there is an Alzheimer's pig model that has been produced, and now we're just kind of waiting to get they're waiting for the pig to age enough so they can start actually looking at it and seeing if it is a good model for humans. So there's a lot of, we're close, and I think it'll take something like that. Once we have a good model for Alzheimer's or cancer or something like that in the pig, I think it'll be a little easier for one of these pharmaceutical companies to kind of jump on board and give it a shot.
And hopefully get us closer to getting a better drug. Because like I was saying, like Aricept is one of the biggest Alzheimer drugs. It's absolute shit, in my opinion. It's also like $200 for a 30-day supply.
Thank you for adding that, in my opinion, because that removes liability from the three of us in case they decide somehow see this as
from my personal research. I mean, and even the real research itself is like, it kind of does stuff, like, and we don't even know exactly how that even works.
It works in 0.00001% of people.
Yeah, we don't. That's not a real number. That's a made up number. But yeah,
totally made up. That's a 5 sigma certainty that's made up.
Exactly right. So it's frustrating because it's like you have such a devastating disease like Alzheimer's. Like I have Alzheimer's patients that like it's very, very sad when you see them, their brains deteriorate, and I have a family member too that's deteriorating from Alzheimer's, and it's like it's really frustrating when they push like drugs like the other ones that are out, and then I don't really see them working, and a lot of the research agrees, and it's like, well, that's bullshit, because then it's going to stymie the research into it, because they're going to be like, well, this drug's not that great, but do we really want to spend millions of dollars to research another drug?
Yeah.
Maybe there isn't a cure, kind of thing.
And the saddest thing about that stuff too is that when they have those moments of clarity and then they're just like gone again. It's just like, oh, hey, how are you doing? Like nothing's wrong, and then all of a sudden it's just like,
right,
who are you? Or
I see it where they're arguing with me about their copay, and I just people like shouting matches. Actually, recently we have a few patients that have Alzheimer's, and they are like really falling apart, like so much that they're like complaining to corporate about stuff that really didn't happen, and like just really scary stuff where it's like they literally cannot function. They're driving and it's like, that's scary. And they're on these drugs, and it's like they're not working. And it's like, well, why are they spending this money? Like you almost want to be like, I don't want them to be on it anymore. It's not. It's too much cost, and it's not that much that's helping people. So hopefully, like research into pigs and further could be beneficial. So my question, like, okay, we have the pig model now. Is there anything one step up from the pig model? Like, would that be dolphins? That would be even closer to us. Like, if you base it on intelligence,
getting that past PETA.
I guess, yeah, right. Well, I guess it would be. I'm gonna fuck this up, but apes. I guess apes are close to us, right?
Yeah, a lot of, and there is a lot of. Well, I won't say a lot, but there is also research that's done with what we just kind of refer to it in general as non-human primates, which can be like monkeys, chimpanzees, that kind of stuff. There is people do a fair amount of research with that, and a lot of not all, but there's a lot of testing that gets done in primates before humans. But one of the main issues is just the ethics of it, and not to mention also you've got similar costs, and you've got even longer generation times for primates usually than pigs.
Longer than pigs. And I was going to say it's going to be a hell of a lot more expensive to have a primate than a pig,
because apes live for decades, don't they?
They can live for 50, 60 years, some of them. So it would be,
and that extends the generation time too. I mean, if you've got a situation where you're looking, I mean, a lot of mice studies, they'll look over generations, crossbreeding mice and different things like that. If you're trying to do something like that in primates, you could be talking years for generation times. I mean, it could take you 10 years just to get the final product to start doing the experiment that you want to do.
And you're talking like generations of actual scientists who would have to be doing a lot of the research. That could get extremely complicated, and funding again.
Extreme.
It could get really frustrating, because there's, I don't know. Can you talk about a little bit of their egos in a lot of the research you have? Like not egos? You. Yeah. Like, do you think like a lot of your fellow researchers have like, negative egos that sometimes affect the research? Because I mean, we're all human, so we all have.
Oh yeah.
We all have
Oh, yeah,
humanistic
there's-
things that can do that. So can you talk a little about that?
Yeah, I mean, there's all sorts of. We just refer to it in general as the politics,
Yes.
which I'm sure is similar to the kind of stuff you would experience, in any sort of company. It's the same thing. There's people
I'll tell
who
you
think
all
that
about company politics if you
know,
want.
they're doing the next big thing. They're the next. They're the most important guy out there, and their research is more important than yours. So they need the space for their animals. You can't have it. It's just like there's all sorts of that kind of stuff, and people try to go backdoor to try to. Like we've got a research facility where we house a lot of the pigs, and there's a limited amount of space, and it's kind of like a first come first serve. And then there's some guy who got money, so he was trying to convince the university that he should be able to reserve like X amount of rooms for the next 3 years, and it's just like no, you can't do that. It's not your. This isn't your personal facility. There's other people that are here. You gotta
Right.
share.
So do you watch Big Bang Theory?
A
CBS.
little bit.
There-
I'm a little behind though.
There was, I think it was in like the second or third season, there was this whole thing where all the funding was getting cut back, and Sheldon was all like, I don't understand why they don't just cut everybody else and just give me all the funding.
Yeah.
I'm clearly the smarter person.
That show does a really good job. Like
Yeah.
that's kind of how a lot of it is. It's all like it's all about me and not the whole common good of
Yeah.
everybody. And it's hard to do that when you're putting in 16-hour days, days and days and days and years and years and years. Like I can understand that you start to think that it's so
Oh,
important.
yeah.
And a lot of it is. A lot
And
of
when
the
it's your
research-
life's work, it's important.
Exactly. Like it's your life's work and
Yeah.
it's your life. So it's sometimes frustrating because it's like you have to balance that personal life, work life, and kind of have to be more grounded. And it seems like you are much more grounded, Kyle, than the average scientist, I guess we'd say.
Well, and I've also I would say I haven't quite been in it as much. I feel like once you've been at a university for 20 plus years and you're tenured, you kind of start to I think that's a lot of that starts to come into play.
You do, yeah. You kind of get a little lazy. And I don't know if it's all your fault. I think a lot of the politics kind of just kills you. That's what I've heard and seen a little bit too. That it sometimes gets really frustrating to go 110% like you do when you're younger. You know what I mean?
Oh yeah.
But
yeah, the only because as an older, well, I wouldn't say at all universities, but at least at the University of Illinois, where it's a research university, you get whatever your salary is. The university pays half of that, and then the other half comes from grants or funding that you acquire. So that's kind of their way of keeping everybody going. Is if you want to get paid, you got to find funding. You got to get money coming in.
now,
Got any tips for raising money?
I was going to say this is I was going to bring that up because this is the thing. Is there any research that goes to like Kickstarter and uses like crowdsource? Because we do have a Kickstarter on Geekify.tv.
Shameless plug.
Shameless plug. I have to. You can
Yeah,
Kickstarter
right,
and find
well-
us. Like, are there are there
Sorry, I'm
universities
coming.
that go to Kickstarter? Like, that'd be a really good idea.
I think you can use Indiegogo, but not Kickstarter. Kickstarter has to have an end product usually.
okay.
Indiegogo, you can use like nonprofits.
Could you
and
make
stuff, too.
like if you donate $1 million, you get like a cure for cancer? Like, like I'm surprised that there's not more crowdsourced funding for a lot of the research. You know,
There
what I mean?
is. It's called income tax.
Well, okay, but a lot of that's funded through we don't
yeah.
get to see that directly
No,
go.
exactly.
Like because I would love to be able to join something and be like, oh, I'll kick in $10 a month for a year for some sort of research project. I mean, that doesn't sound like a lot, but like let's say if a few million people did that,
Yeah.
you could have a lot of funding. Like I'm kind of surprised no one has started that.
Yeah,
Copyright
you could get it.
Geekify.tv right now. Okay, no, but
We're gonna patent that.
exactly, because I think that might be a good way to go. Because crowdsourcing is a great way to do a lot of things
Yeah.
nowadays. Speaking of, we just found this one really cool. Brett showed me before the show. This one, it's board game.
It's
okay?
a D&D clone essentially.
It's a D&D clone, and they had it up there. They wanted to get $35,000. Okay, pretty decent to make this D&D clone to sell people. They made over $2 million on Kickstarter.
and it was a 60-day project.
60-day project, yeah, $2 million, and they
With,
only
like-
really wanted 35,000.
With like 5,700 people that donated.
Yeah,
Yeah.
it was insane.
And
Wow.
and he showed me this, and I was like, "How the fuck did they get that?" I mean,
Yeah.
good for them. I'm happy
Yeah.
for them because holy shit. But
it proves that the system can work.
yes,
Yeah.
and it has to be something that catches fire. And I think it'd be very cool if we could do that in the research. I'm kind of surprised drug companies don't kind of do that too. I know you get.
Oh, you want a cure for Alzheimer's?
That's
Here
what,
it
yeah,
is.
exactly. They can't do that.
That's
but
exactly
like,
why.
some sort of like humanistic way that we can have people that donate to it that is more transparent than like an organization, you know, that exists now. You know, just something like that would be really cool to see. I think a lot of the problem is too that a lot of people don't have money.
Yeah. Especially now.
Especially now. Yeah, the economy is kind of tough for a lot of people, but I mean, Kickstarter seems to be doing well. Didn't they just pass a?
Yeah, they made it somehow easier for companies to be able to take in crowdsourced funding with some type of tax revision or something, where it made it easier for startups to use crowdsourced funds.
Which, you know, it'd be really cool. It'd be really cool if they did some sort of research thing where it was like people donate X amount into research, and knowing that it may or may not get anything great, but just to have another way to make money, because I know, as you know, at the university, it's really sometimes tough economically to move on. You know, it's just sometimes you have to muddle through that bullshit and delay projects because of money. Now, Mike, I have another question for you. Actually, for have you looked at a lot of how Europe does their stuff? Because I know theirs is a little bit different than how we do it. How we do research. I mean, like, have you looked at how they do a lot of their funding and stuff like that?
How they do the funding, I don't know a lot about the funding. I mean, I know they have similar, you know, you've got similar ideas with the NIH and different outside groups that will provide funding. They also have a little more unity, especially when it comes to funding for students, like for postdocs or PhD students. They have a lot of funding that, well, I can't access because I'm not a EU citizen, but they've got tons of they put a fair amount of money into it. I wouldn't necessarily say it's more than we have here, but there's a lot of you know, that's a great thing about this field is it's everywhere. And you know the U.S. is pretty strong. We've got some really you know, some really impressive biotech companies, pharmaceutical companies. But Europe's also doing pretty decent. China is huge when it comes to sequencing. I mean, they've got facilities over there where you can supposedly sequence an entire human genome in a day.
Yes, I've heard about some of those, that they have some really, really incredible, like the frontier technology in China. Yeah.
Yeah, so it's basically they have these machines, which university has one on order, maybe, or maybe we have one, but they have these machines. But basically, what they do over there is they just they build warehouses and they just fill them with these machines, and they just have, you know, staff that goes through and puts in the different things, and it's just like, yeah, they can pump out data like no other. I mean, it's unbelievable.
Right, with unlimited funding, I mean, we would have a cure for everything, you know, because it's
unlimited
out there.
funding would have been on Mars in the '60s.
Exactly,
Yeah.
and that's part of one of the big barriers, I think, is funding, and it's I think it's really cool, like you say, it is everywhere. I mean, Canada has their own research, and everybody does it a little differently. Like everyone's doing it, just they have a little tweak here and there. Especially like with my background with drugs, I see that a lot too, where Europe will approve drugs a little faster than the United States. Like for the same drug that's coming there, they're sometimes a little bit better at doing that. And
Socialized medicine there though too. So
exactly, yeah. There's a little bit of that as well. It's a different system to do the same thing, to get drugs to people. So it's always fascinating to see. In the research world, I guess it would be the same. It'd be really cool to see the little differences, the nuances from here to there as well.
Yeah, it's totally different. Just the culture. I mean, the culture of the people, but also just the culture of the workspace. I mean, they talk about. It was a cool thing about being able to go over there and doing that class that I did. You get to experience what other people's grad school, careers are like, and it's quite a bit different than it is here. The courses are laid out a lot different over there than they were for like my PhD program. Stuff like that. But even stuff as simple as in the U.S., if you're at work a lot of times, if you're working, you probably working at the computer. Maybe you eat your lunch at the computer or have a snack or coffee or whatever, while you're working. That's something in France especially, but most of Europe, they don't do. It's considered antisocial, for you to sit at your desk like through lunch. They actually, everybody takes a break and goes and has lunch together and talks. And
I wish.
I wish we could do that here. Right? Yeah. We have a little bit. I don't want to say stronger work ethic, but we have this like driving work craziness. You know what I mean? I suffer from myself. You know, working a ton, and sometimes overworking yourself a little bit can get you in trouble. And yeah, Europe does have this where they realize a lot of those social things that you need to just you know, take a break and do this and do that, and the work will get done, kind of thing.
35-hour, 30-hour work week, and get a whole month off every year.
Yeah,
kind of good stuff.
The vacation's unbelievable, man. Yeah, I was talking to one of the grad students over there in France. He asked me how many vacation days we got, and I was like, oh, we get 12, which I think is we get like 12 a year. And he's like, really? That's it? I'm like, wait, how much do you get? He's like, oh, we get like 32 a year.
Oh my god!
Wow.
I know.
I can't really say anything because the advertising industry always has ridiculous vacation. Like, I have more vacation than I know what to do with usually.
Lucky.
I would get like
Wish.
10 personal / sick days, 2 weeks of vacation. Between Labor Day and Memorial Day, we get an extra 5 days to take off just any Friday that we want, and then we're closed for every federal holiday, and we shut down between Christmas and New Year's.
I got 0 6 days in pharmacy school.
Yeah,
all through, even the 4th year, which is your clinical rotations and stuff like that. They're like, oh, you don't get any sick days. Do not get sick.
Yeah. You're around all these sick people, but do not get sick.
Yeah, if you do, you have to make it up somehow. Yeah. You know, and hopefully it's not like you're in the hospital or something like that, and you have to make it up like that. But yeah, so I understand that. It'd be very cool if we had a few sick days just to make you feel a little more relaxed sometimes. You know? Yeah. Because then it makes you more antisocial. You're like, I don't want to go out to that party because I don't want to have the potential to get sick.
Yeah. Or I don't want to be hungover.
Exactly. You know,
hangovers are never a problem though.
No,
never.
For you, maybe.
That's just a state of being in the advertising industry, especially on Fridays.
Yeah, right. It gets like that. So, do you have anything else to share with us about science, Kyle?
Actually, I've got a question.
Uh-oh.
I brought in a question. Look at me. What? So, what exactly do you hope to do with your postdoctoral work? Like, what's the difference between actually getting your PhD and then, okay, I'm doing my postdoc? I mean, what do you benefit from doing postdoc work as opposed to getting a job at some corporation doing something? This is just the capitalist in me coming out. So
yeah. Well, no, so postdoc is basically just a nice way of saying that a PhD is not enough for you to get a job anymore.
Gotcha.
If you want, I mean, I could probably go get a job at like pharmaceutical company or industry or something like that, working as kind of like more like a worker bee. But if you want to be conducting your own experiments and and working in research like outside of a biotech company, this is really kind of the route you have to go. So a postdoc is basically in between a PhD and a professor.
Gotcha.
You're basically doing the same thing. I'd be working on a project which depends on the lab that I get into. Hopefully, it has something to do with my PhD. Like I'd like to stick with pigs. I want to stick. I really want to stick with biomedical research. I think that's really interesting, and the bacteria host interactions that I've been doing with have been pretty cool. So I want to be somewhere in that field. And so it's like a 2 to 3 year position. And it's basically like the same thing. I'm doing like another project, but I don't take any classes. And I probably would have a couple PhD students that would be working on the project with me, or I'd kind of be overseeing.
Slaves, whipping boys.
Exactly. Looking forward to that.
Yeah, you basically want to be the boss. You don't want to be the grunt. You want to be sitting in your nice little chair. Yeah, I don't blame you.
Yeah, I'd like to see, because I want to. I'm not opposed. I've been in academia, my whole life. Obviously, I've never even had a real job. Basically, I've been doing nothing but study. So, I love the university setting, but I've never experienced the industry. So I would like to maybe work for a biotech or a pharmaceutical company for a while. But I could see myself ending up at a university. It's just so much. Well, it's as stressful as you make it.
Gotcha. Right. It's different. It's a different beast in academia. Yeah. Yeah. I never really wanted to be in academia during. I didn't really like the academia side of pharmacy. At least that's what I was exposed to. So I was definitely like, I would rather be working in a pharmacy somewhere in retail. Is what I like the most.
Okay.
So that's I can understand. Like it's you want to. Sometimes it's nice, but hopefully your research is a lot better than pharmacy.
It's not boring being in retail, that's for sure.
Yeah, no, I get exciting stories all the time.
Every day it's something different.
It is
awesome about it.
Well, yeah, it's exactly. It's exactly. You work in retail, so you understand.
Yeah.
The same thing. It's always like someone comes in. What the hell is going to go on now?
Yeah.
And if you like that, then it's great for you. You know.
Should I answer this question or should I toy with them? So
Oh shit! They came in all pissed off. Let's fuck with them.
Yeah, exactly.
Way worse.
Yeah. So yeah, it's just two different sides of the same coin, which is really cool to see.
Yeah. Yeah, pretty much.
Well, I'm happy for you, man. I mean, congratulations on everything. Good luck to you with this. It's exciting stuff. Definitely keep in contact with us. Hopefully, have you on again.
Oh
do
yeah,
a little bit more research.
Preferably when you're in the Netherlands or something, so it can be like 3:00 in the morning. Tell Lisa to go away.
Yeah, I know. Right?
Yeah, we can give you when it's 3:00 in the morning. You're all groggy and trying to drink some coffee and talk some science.
Wait, no,
with
he
you
can't
again.
have coffee. He'll be antisocial then.
Exactly right. Yeah, he has to have somebody else.
No,
there.
wait. If he's in the UK, he has to have tea.
Oh, it's true. You do have to have tea there.
Oh my God, yeah. Yeah, if you ask for coffee over there, man, I've made the mistake before, and you're better off with tea. That coffee is just no good.
Right, and there's still caffeine in tea because you're in school, so you have to have caffeine.
It's not espresso levels of tea, though.
Yeah,
That's what I
just
feel
not
my
Starbucks
body
crack
with.
levels. Yeah,
that's what. Yeah.
Two triples every day.
Yeah, right. Oh my God, your heart's gonna explode. Well, thank you again for being on the show. It's fascinating research. You're doing God's work, I guess you could say.
You are God.
Yes, you are.
I will say it.
You're God of the porcine community.
Yeah.
That's awesome.
Ooh, I'll take it.
Exactly. Well, good luck to you, man.
Great. Yeah, I've enjoyed it. This has been a lot of fun.
Yeah, it's good stuff. It's fun to talk science. It's good to get a little bit different from our other show, 3 Guys 1 Cup, and talk a little bit more like actual science down. And hopefully, some people find it entertaining. I think some people will. And
yeah. There's the community there.
Yeah.
So
there's something for everybody with us. So, well, thank you again for coming on, Kyle. It's been awesome.
Yeah.
Any final parting thoughts for us?
Just everybody who's listening. Hopefully, there's a few people out there. Just like
5
to say
at the moment. 3 of them are
what?
us.
Yeah, right.
So for the 2 people out there, right?
Yep.
Thanks for having me on, and make sure don't believe everything you hear. Actually, look into the science and look into the research of it, because you'd be amazed at how easily it is to skew data or make something not so true seem.
Look for the 3s and 4s. That's the sign of bad data.
It's funny you say that. I want to have a whole show. Actually, I was thinking before of just that, of like going into why how you can take something and completely change what it really means. And you're right. Yeah. You always should be challenging something like that. Like think for yourself, challenge authority, kind of thing. Always. Don't
read an article and just see it as fact. I mean, because they're people just like you and me, and they don't always know what they're talking about.
And we make mistakes, and sometimes people can get biased, even if they don't realize it. There's a lot of that, a cognitive bias that happens in there. So yeah, definitely, that's good advice. I like that. Yeah,
definitely. Actually, it happened recently with there was somebody in Europe who used rats to try to prove that genetically modified corn gives you cancer.
Yes, they did, and they used the cancer mice.
Yeah, and they used mice that are prone to cancer. They were like, oh, okay.
Well, there's your problem right there.
Yeah, I did hear about that, and a lot of people were really pissed off when they looked into the research, and they're like, "You jackass! You were using these cancer mice. Like, what are you doing?
Well, anything you give them, give them something, they will get.
Yeah, give them water, and it's going to cause cancer. Yeah, there's definitely that bias there.
You'd be amazed at how many times I've seen that paper cited for people that are anti-GMOs and that sort of stuff.
Right.
I mean,
well, going even bigger than that, look at the paper of the guy who did the autism study with vaccines.
Oh yeah, I remember that.
He has come out and said that all of his research was wrong, that he lied, like he flat out made up data and lied. And look at the damage that's caused.
You know what that guy's doing now? Mopping floors.
Yeah, right. Yeah, I think he was in Europe actually. I think he was just completely lambasted. Like they were just like, he's done when that came out. But the damage is already done. It's very hard to reverse a lot of that.
Yeah, it's too late. I mean, people have already cited it. It's already, as far as other people are concerned.
Jenny McCarthy believes in it, or Jenna McCarthy, is it? She's crazy.
Oh, Jim Carrey's former lover.
Crazy about that shit. Yeah, exactly. So it's already, the damage is done.
Yeah.
So
yeah.
Yeah, you got to challenge everything. But all right, thank you again for having me on the show, man. I appreciate it.
Yeah, stand by while we wrap up here, because we'll talk a little bit afterwards. But
yeah, definitely.
This has been the first episode of Science.
And it
Finger
won't be
to the sky. It will not be the last. I don't know if this will necessarily be a weekly thing, but monthly at a bare minimum.
Yes.
We'd like to obviously do it more frequently, because we all are science fanatics here at Geekify TV, which is kind of just falls into the line with our culture here at the Fortress of Davitude.
That's here.
That's here.
Yeah, we are gonna get more signs on the wall. I know the wall looks very blank and sad right now, but we are gonna fix this place up a little bit. You know, bear with us. It's a growing experience for all of us here.
Yeah.
Yeah.
You can follow the Science Show specifically at SCIENCEEE with 3 E's pod on Twitter, Geekify TV on Twitter and Facebook. And please find us on Kickstarter. And if you like what you're seeing and all of our other shows that we have going on, please back us. We're trying to upgrade our equipment, which we're slowly but surely doing. Every show gets a little bit better and better and more professional quality, I feel like. We're getting there. We're learning the video editing software and all this kind of stuff. So we're trying to really make this into something, and anything, a dollar would be helpful to get us to our goal. We have 38 days left, and we're 18% to goal. And there's a really high statistic of people who get over 20% to their goal almost like 80% of the time then actually make their goal. So we need that close. 2 more percent to make sure, we're going to get this.
You know, we have a ton of shorts coming. We have a ton of ideas going for other shows.
Like, we upgraded our filming stuff for the shorts.
Yeah, we got a little bit better camera work, and we're getting used to the technical stuff because none of us have a background in this. So we're all just figuring out like just stupid children playing with toys. But, you know, like, it's a slow process, and we're working to get better. We're always trying to get better. We're not just going to release crap all the time. We promise. You know, but... just bear with us with a lot of this. It's a lot of fun, actually.
Yeah, I enjoy this.
It's a lot of
I
work
look forward to
sometimes.
this every week.
Right. It's good stuff. It's good. It's just slowly building this up, and hopefully we can build up an audience that would keep coming with us and everything like that.
It's by far the fastest hour of the week for me.
Yeah, definitely.
Yeah, very much so.
Definitely. So, all right. Thank you, everybody, for listening and watching up on live stream and stuff like that. I'm Dave Mankoff.
I'm Brett Kaminsky.
Andrew Nopinski.
And you are... Kyle.
You on the screen. What's your name?
Hello, sorry, Kyle.
Whoa,
Kyle.
Zoom.
Kyle, and how do you pronounce your very German last name?
How do you pronounce it? It's Schachtschneider. It's much easier if you just don't look at it.
I'll try not to. I'll just think about it.
Just don't look at it. Just say something. Blah, 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 blah.
Exactly. Just say something very German sounding.
Yeah, that works.
Awesome.
Enough.
Well, thank you, man, for being on again. And we'll be back on this Thursday with 3 Guys, 1 Cup. Thursday, I think about 8, 9 o'clock or so.
Yeah, probably about 8.
Probably 8:30.
We'll let you guys know.
We'll figure it out.
Yeah, we'll let you guys know. All right. Well, thank you, everybody, for being on. And I guess that's it for tonight.
Have a good night.
Bye-bye.
Adios.